And we all knew that the invention of microscope follows closely behind the invention of telescope in the early 17th century. That is, follow Galileo, who put together two lenses and then constructed a telescope. He looked at the sky and found that, oh, the moon is full of mountains and basins. And then he trained his telescope to look at the sun, to look at the Mars, to look at Jupiter. He found so many things. So this is a marvelous world. That is newly discovered world. And for some people who has been able to grind very small lenses and put together a compound microscope. And this compound microscope is able to look into very small things. So this rubber hook, the first rubber hook, who made the compound microscope, look at the bark, look at the cork and discover the cell. That is the empty space in the cork. They also look at the mold and discovered the floating bodies of the mold. So this really opens the microscopic world for human investigation. Then around 1666 or 1667, a Dutch haberdasher is the cloth merchant, who is able to grind very small lenses. And his microscope is able to magnify about 400 times. And he looked at a drop of water, a drop of river water, or a drop of sewer water, and discovered many small animals swimming. So this is called the animal cures. And this marvelous discovery was reported to the Royal Society of London. That is the most important British science society. And there was his observations were published. I think, but from about 1660s to about 1840s, that is 1.5 centuries. The ability to make microscope is still restricted to very few persons. So up until the middle of the 19th century, can people buy factory-made microscope and use that to observe small things. So starting at about 1847, there is one doctor called Ignaz Semmelweis in Austria. Who he is a surgeon. He's a obstetric surgeon. That is, he works on the obstetrics. That is, taking the babies. That is, helping the baby mothers to give birth to babies. And during that time, the puerperal fever means the fever on the birthing bed, and many mothers get infected and get severe inflammation and died. So the death rate is very high. So this Semmelweis, although he's not able to do investigation, but he heard of the series of the germ theory. The germ theory means that the disease may be caused by microbes, by very tiny microorganisms. So with that in mind, he instituted washing hands for all the doctors in his ward, in his obstetrics ward. And he can drastically reduce the death rate of the mother. So he proposed that the washing hand and cleaning, hygienic procedures is very important. But he was ostracized, that is, was severely criticized by his colleagues. So in disappointment, he went back to Hungary, and then incidentally got infected by the same bacteria of the disease, and so he died. But this and some later usage of the phenolic acid, carboxylic acid, that is about 1867 by Lister, Joseph Lister. He's using the phenolic acid, the carboxylic acid. Carboxylic acid is actually the water solution of phenol. Spray it on the surgical wound to reduce the infection. And he is also able to reduce the infection dramatically, so as to save a lot of surgery patients. So these two, that is before the germ theory, before the confirmation of the germ theory. So even the proposal of the germ theory, that is barely a theory, some doctors, practitioners is able to use that hypothesis to do something good. That is to apply this hypothetical knowledge to the care of patients. So that means that even with proven, that is a hypothetical theory, is of some help to develop some techniques, and these techniques is able to save lives. And during the later part of the 19th century, people saw the first blossoming, that is flowering, of medical microbiology. So there are two giants in this field. One is the Louis Pasteur. He has two major discoveries among many of his discoveries. That is, he first repudiated, that is falsified. Falsified means that proven it wrong, proven the theory wrong, of spontaneous generation. The theory of spontaneous generation is that since God created the universe, some remaining living power. This living power is able to make non-living things life things. That is, if you leave broth under air, a few days, the broth turns sour, turns rancid. So that is originally the soup. There is no bacteria in it. Then suddenly there are microorganisms in it. So this is called the spontaneous generation. And this theory was at that time believed by many experimentalists, because when they leave some broth, some nutrient broth on the table, and then it will invariably give rise to tremendous amount of microorganisms. So this is experimental truth. But Pasteur thinks that there must be a seed from the outside. So in non-living things cannot turn itself into a living thing. So he did this experiment, the swan neck experiment. That is, you can see that he has a flask, but the neck, he's turned it into a swan neck. So the purpose is that this swan neck can connect to the outside fresh air. However, if there's any particle in the air, it will settle down in the middle. That is the lower part of the swan neck, the neck, and because it will not rise and get into the flask. So his swan neck experiment is so successful that he has actually demolished the spontaneous generation theory. Then we can only know that microorganisms also has its origin, and this origin cannot come through spontaneous generation. And later on, he has many other many other discoveries, such as discovery that the wine fermentation is through the yeast, and other kind of fermentation is through other kinds of bacteria. And so his contributions is tremendous in the later part of the 19th century. Another German scientist is Robert Koch. Robert Koch, he's a country doctor. He likes to do research, and he is able to first grow bacteria. Especially, is able to show that anthracis of in animals is due to Bacillus anthracis bacteria. And this through the study of this bacteria, he formulated the Koch's hypothesis. Postulate. Koch's postulate is that how do we prove that an organism, microorganism, causes a disease? That is, first you are able to isolate the bacteria from this sick animal, and then you should be able to grow the bacteria in pure culture. Pure culture is that the culture, the growth of bacteria outside of the animal body. And then if you use the pure culture to inoculate into a healthy animal, the healthy animal got sick again. And then from that animal, you are able again to isolate the same bacteria. So this is called the Koch's postulate. That is 4 steps. 4 steps of the experimental manipulation, which can convince people that this bacteria causes a disease in this animal. And he is able to do it first in anthracis. Anthracis is the kind of black death in animals. Then he is able to show it in tuberculosis. Tuberculosis is in the late 19th century, in the early 20th century, is one of the worst infectious disease in the world. It kills about many people, about 17% or 20% of the death annually in the world by this tuberculosis. The problem with tuberculosis is that this bacteria grows in the lung. However, it has a thick membrane, thick lipid envelope. And this lipid envelope is difficult to stain. So he invented a method to stain it. And this method is later on improved by other people, and this is called acid-fast stain. Acid-fast means fast, it means resistant, acid-fast. That is resistant to the acid. Acid-fast generally is that you have to boil, to heat the bacteria, the specimen of bacteria, and using the acid. This acid will facilitate the dye to get into bacteria, and then you use the acidic alcohol to wash it away. So this way it can stain the lipid-rich tuberculosis bacteria. And he's able to show the presence of tubercle bacilli in the lung tissue, in sputum. Sputum means it's the coughings out of the throat of the patient. It's a sputum. And to show that it does exist in the tuberculosis patients. Since this discovery, so people are able to cope with tuberculosis in a more effective way. That is, try to not get infected by tuberculosis bacteria. And so from about 1880 to 1910, so these 30 years are shared in the golden years of the medical bacteriology. medical bacteriology. And during this time, we can first see that for the Oriental scientists to participate in this golden opportunity is only the Japanese. So we can see many Japanese names in the bacterial name. That is, they had participated in the discovery. That is, they went to Germany and joined Dr. Koch's laboratory, and they are able to discover the pathogenic bacteria in many different kinds of infectious diseases. So this means that we Orientals are able to really get into the science and the help the growth of science. However, not many other people from the Asian society is able to participate in this golden chance. Okay, now let's look at the second picture of the history of microbiology. So this is actually the development in the 20th century. In the 20th century, it is marked by the fusion of genetics with microbiology. That means that microbes or bacteria were used as a tool for the study of genetics. So this started with the Mendel, and the later part of the 19th century and the early part of the 20th century is that is the genetics is gradually being studied using the bacteria. And then starting in the 1928 is the discovery of transformation principle. Transformation means that you give a piece of DNA to bacteria, bacteria will take up that DNA and transform its own phenotype. So this transformation phenomena was very important. And then some people jump into the microbiology, especially bacteria study, in order to understand the genes. And then there are many discoveries, that is, such as Beadle and Tatum, Griffith, and Avery, MacLeod, McCarthy, and Shargarth. And so all these investigations from the 40s to 50s actually stimulated interest in DNA. And this the discovery of DNA structure by Watson and Crick actually started the golden age of molecular biology. Golden age of molecular biology. That is, once the DNA structure was proposed, and then people are able to think, wow, DNA is much more important than proteins. DNA has the genetic material. So it started the golden age of molecular biology. And so the basic process of replication, transcription, translation, all these that is central metabolic processes, non-metabolic processes, central information transfer processes were discovered. So this is really exciting. And then from the 1980s, the focus again shifted out from the bacteria back into immunology, into cell biology, virology, cancer research, and so there are tremendous progress made in the next half of the 20th century. Okay, then if we look at the next PPT, history of microbiology, then we can see that the age of genomics now appeared. That is the sequencing of DNA made possible by Max and Sanger, the method. these simple tricks were combined with computers to make the second generation sequencing machine or the third generation sequencing machine. First generation sequencing is through the gel, which I did when I was a PhD student at Harvard. It's a very difficult process. We have to wash the plates, make thin gels, and then run the DNA sequencing on the very thin gels. So it takes a few weeks to get some sequences. But this process was computerized. And then in the early 21st century, the next generation sequencing appeared. So one machine may generate trillions or billions of DNA sequence in a day. So it's really revolutionized the sequencing. And so the development of the past 20 years is so tremendous that it ushers in the science of genomics or proteomics, all sorts of omics. That means that they are looking at the DNA sequence, looking at the RNA sequence, and looking at the protein in the whole holistic view. That is, the whole bacteria, whole animals, whole organisms. That is, they can process billions, trillions of information using the computer. So this is actually a tremendous progress. However, the progress is so rapid that it is almost difficult for the peripheral societies to catch up. That means that even for me, I got my PhD in 1982. And in the era of first generation of DNA sequencing, and also the molecular genetics era. And so I am totally oblivious to informatics, to genomics. That is, all these sciences are new to me. And of course, they are a must for you. So looking at the past few hundred years of the microbiology, we can see the tremendous progress in the science and in the technology of microbiology. However, for us, the Eastern societies, to catch up, I think it's even more difficult. That is, we lag behind even more, even faster than those Western societies. So it's really important for us to try to find ways to catch up with the progress in the Western societies, and we have to devise ways so that we can disseminate the knowledge, disseminate the technology to our own society, so that we can generate many future generations of microbiologists, and they can take up the task of promoting this science, and so to explore new field, that is, make new discoveries in this field. Okay, so this is a brief introduction to the history of microbiology. Okay, my purpose is that by looking back at the past achievements, we have to think how we can handle the future work of this field in our own societies. Okay, so let me take about 10 minutes rest for you and for me, and we'll start again at 9:50. Okay, thank you very much.
Okay, sir.